All right, I'm back and I've got something to show you that's kind of unique right here. I have a video of water. Not really a video. This is a computer simulation, okay? But we are going to be able to show you what the water looks like by the computer simulation of it. The computer is taking all of these water molecules with each of their magnetic dipoles and each of their hydrogen bonds. And remember, a hydrogen bond is just minus, plus, minus in a row. Each of these is shown. When a good hydrogen bond is formed, it shows it inside the water. It shows it at the as a dotted line. Okay, so I just want you to watch and see what happens as we press play on this, and the computer lets the water move around. You see that there's a lot of motion. This is actually cold water, and so water at room temperature is even moving around more. But you notice how the hydrogen bonds are really holding it together and making it sort of sticky to itself. It makes it stick to itself. And so you can see them, they're still able to move around. This is water on the verge of freezing. So water can move, but its hydrogen bonds are tight and hold it together. I just have to show you a few pictures of what water moving looks like. This is where you can find the video on YouTube if you want to watch it again. Now there's scientists who study this, the scientists who made this simulation of this video. They actually will study the shapes of the water, and it turns out that because water sticks to itself, it forms very complex structures that form and break apart. They form and break apart. And these structures determine how it works and how other things dissolve in it, how water sticks to itself and how water sticks to other things. And so I just wanted to give you an example of the kind of things that they do. And I think it's also kind of a beautiful set of structures. Water makes a lot of pretty shapes when it comes together like this. And each shape lasts for only a couple of nanoseconds or maybe milliseconds. But then it moves on to another shape, and water is a constantly changing, constantly flowing, but always close together and always fairly tightly bound kind of substance. These tight hydrogen bonds define what water can do. And this actually has an importance. If you look inside insulin, here's a picture of the structure of insulin. Remember, insulin is a short, small protein with a number of protein chains, and it's held together by zinc. And if you look in the middle of it, there appear to be some gaps in the insulin structure around here. Now we know that those gaps are actually filled because everything gets filled in a liquid. The liquid will find its way in. There's protein side chains, of course, that are fitting together with each other, but they don't fit together perfectly. And water will actually get into the middle there, and it will hide in the middle. And in fact, the water will adopt these cluster shapes inside the protein. They found where the water fits inside these holes in insulin. If you zoom in close to the zincs right here, you can see that there's a lot of waters, and they're even forming shapes like what I showed on the last slide. This is a whole paper about it, but to really get into it, it's really physical chemistry beyond the scope of this class. But I want you to see the water molecules stuck inside the protein, and they're actually important to give the protein shape. And we are still figuring out how they are important. And so I want you to see that there's a lot to learn about how water works. Here's another view of the same cavity. You can see the waters, and you can see these little green hydrogen bonds holding them together, like I think it's right up there above my head. If you take away the water, the insulin, by the way, becomes much less stable. So remember, because hydrogen bonds can happen between any properly arranged electronegative atoms, between any arrangement of OH and O, or nitrogen as well, oxygen or nitrogen, They can happen between any two molecules that have atoms that fit that example. And so, for example, I've already talked about the example on the left. They can happen between water and a hydroxyl group on an alcohol. Move over one, it can happen between water and a ketone group. But then, of course, proteins are joined together with amide bonds, which have carbonyl groups in them. And so water can form to them. Water can also form to the amide. And it turns out that you don't have to have water at all. If you get the arrangement, you can have one amide bonding to another atom. That's the third example on this. Water can form, or not water, but hydrogen bonds can form between peptide groups in polypeptides. And then, of course, you can even have it in DNA. DNA is basically arranged so that you form very good hydrogen bonds between the bases of DNA. And this is one of the ways that DNA works so well. You have hydrogen bonds which are strong, but not quite as strong as a covalent bond. Strong enough to hold the DNA together, but weak enough that when you need to pull the DNA apart, you can pull it apart and it will break the hydrogen bonds. Notice that all of these arrangements have an oxygen or nitrogen, a hydrogen in the middle, then an oxygen or nitrogen on the bottom. That's how they work. We're not going to talk so much about structure, a little bit about structure, but the most important thing is that when you have a hydrogen bonding group, it can hydrogen bond water, and it will help that molecule dissolve in water, because dissolving in water is really just attaching to water. So here's an example of all the different molecules, and hopefully you've seen structures like this before in your classes. When we have the different sugar molecules, for example, glucose right up here. Glucose has 6 different oxygens in it which have the potential to hydrogen bond. All those oxygens are on the outside of the molecule, so water can attach to it in lots of different ways. So glucose is soluble. If you move up and down that column, you can see other molecules that are also soluble. Glycine, aspartate, lactate, and glycerol all have OH groups, which are partial charges, or they can have full charges. Just remember, if a partial charge can help you form a hydrogen bond, a full charge will even be stronger than a hydrogen bond. So that means that glycine, for example, by itself, glycine is very soluble in water because it has the blue groups are very polar, and the polar groups interact very well with the polar nature of water. They form hydrogen bonds, and they form them strongly. On the other hand, move to the left side, or I'm your left, my right, right? So move to the right side of the figure here, and take a look at the typical wax. Wax only has a couple of oxygens in the middle. The rest of it is CH bonds. Those cannot hydrogen bond. So a wax has lots of carbons that cannot dissolve in water. A wax is made up also of fatty acids. And so fatty acids have the same kind of, they would be yellow on this plot right here. They are hydrophobic, and therefore a wax is insoluble, not soluble in water. Then you get combinations of the two. When you have something that has both yellow nonpolar groups and blue polar groups on it, then it depends on how much it will dissolve in water. It will have part of it that will dissolve well and part of it that won't dissolve well in water. So phenylalanine is an example of an amino acid that is amphipathic. Phosphatidylcholine is an example of a triacylglyceride, or this actually is a membrane lipid. It's a lipid molecule that has 2 fatty acid chains that are not soluble, but then it has a phosphate head group that is very soluble. So the question is, what does water do with these things? What happens when you have something that is nonpolar interacting with water? Because the water cannot hydrogen bond to it. These are half soluble, So there's also solubilities of gases in water. And if you look up and down, this is actually on the right side of this figure, you have the numbers for the solubility in water. How many grams of the gas can dissolve in a liter of water? Let's start at the very bottom, right above my head, with hydrogen sulfide. Hydrogen sulfide actually will dissolve 1,860 grams will dissolve in a liter of water. So basically 1.8 kilograms of hydrogen sulfide will dissolve in water. If you look at the structure, it definitely has a lot of hydrogen bonding potential. There are a lot of ways that the SH groups can bond with water's H2O groups. The molecule itself is polar, and so it dissolves in polar water. But as we go up, we get to gases that are important for life, and they are not as soluble. So notice that oxygen, even though it has oxygens in it, the oxygens that it has in it are not, they're electronegatively balanced. There's not a dipole associated with them. And so the oxygen atoms in O2 cannot interact very well with water because they don't have the same electronegativity that water bound to hydrogen has, that oxygen bound to hydrogen has. Oxygen bound to itself actually doesn't have a dipole. It doesn't have an unequal sharing of electrons. And this is reflected in the fact that only 35 milligrams of oxygen, O2, will dissolve in H2O water, 1 liter of water. So 35 milligrams in 1 liter of water, for people like us that are made mostly of water but need to breathe and live on oxygen, this is kind of a tricky situation. What do we do with this? And so the answer is that we have hemoglobin proteins which help bridge this gap, but that's another topic for another day. Right now I just want to explain why oxygen does not dissolve well in water, because O2 molecule is non-polar. So these are insoluble, but something that is polar, like water, has an unbalanced sharing of electrons. Those are soluble. Mostly I want to look at, because we're going to be talking about membranes and lipids, I want to say what happens when a lipid gets dissolved in water. The simple kind of lipid is a waxy molecule, like what we showed on the previous one as a wax. Basically, carbon-hydrogen bonds all over the place. That means that if you have two nonpolar molecules in water, one of the things that you'll see happen is you'll see what happens is like what's in this beaker over here. The oil will actually group together. The oil will come together, and the oil will appear to bind the oil. Okay, so it is energetically favorable for water to pull apart from oil. And if you dissolve oil in water, or if you've seen a small oil spill on the side of the road, you'll see that the oil is floating on top of the water because the oil is collecting together. This is very odd behavior, because what's making the oil come together? What's making this behavior happen? So we need to explain what's going on at the molecular level. Here's an example of before oil groups together on the left, and then oil groups together on the right. We know this happens, so we want to explain why it happens, showing the water molecules. The thing to notice the water molecules that I've circled right here. They are between they're next to the nonpolar molecules before they group together. After they group together, those water molecules have gone off in different directions. They are no longer next to a nonpolar molecule. And think about what is the difference between left and right for those 5 molecules. When the 5 water molecules are next to the nonpolar molecule, they cannot form hydrogen bonds to the nonpolar part. So therefore, they are actually able to move less. That's why I say here they are more like ice. They are more frozen in place because they cannot form hydrogen bonds to the wall of oil that is next to them. But when the oil comes together, then the waters actually are able to escape, go out into solution. They're able to spread out into solution, and they're able to turn in any direction because there's other waters all around them. So there's hydrogen bonds all over the place for these 5 waters. The important way to understand why oil groups together in water is not to look at the oil, but to look at the water. The water is moving out of the way and into solution, forming more hydrogen bonds, not even better hydrogen bonds, just more of them in more directions. So that means that the 5 water molecules are more stable. They are lower in energy, and this is what actually brings the 2 oil molecules together. If you've talked about entropy in other classes, entropy is just a measure of how spread out is the system. And there's a bit of a puzzle because how can 2 oils coming together, entropy should always increase. But you see 2 oil molecules coming together in water. How can that happen if everything should always spread out? The way that it happens is that waters actually are able to spread out more. If you consider the waters are increasing their entropy. We don't talk about entropy too much. I'm a chemist. I have to mention entropy at least once, and so I'm going to mention it here, but it is not the focus of our instruction. I just want to mention it so you can make connections depending on what connections are available, okay? This is called the hydrophobic effect, and what it means, if you have 2 oily surfaces in a water solution, they will come together. The hydrophobic effect is what brings hydrophobic surfaces together in solution. What it really means is it means that water that has been trapped next to that surface before is now freed to be able to hydrogen bond other waters more. It's a little tricky because you've got to keep track of these waters in your head. So it appears to be an attractive force between oily surfaces, between lipophilic surfaces. But the oily or lipophilic surfaces actually come together because the water is being released. That's shown in this figure as well, which is just another version of the same figure that I just showed you. It's just showing that it will happen for an oily or lipophilic substrate and an oily or lipophilic enzyme surface. They will come together. You will actually see the frozen waters next to oily structures. You will actually see that. I've seen it with my own eyes looking at protein structures. So it's important to know that this is real. The water is actually more ordered next to an oily surface, but this is actually bad for its entropy. When it's released, it becomes less ordered, and that actually helps drive things because things become less ordered over time. That's what entropy says. Again, I'm not going to talk that much about entropy, but I'm going to mention here the entropy of the waters is increasing, even if the entropy of the oily molecules is decreasing. And the entropy of the waters is actually increasing more and driving the interaction. Okay. So that's your chemistry, but the important thing to see, oil groups together in water because of this effect. Amphipathic molecules are actually, so this helps us understand what happens when you have a molecule that is part oily, part lipophilic, and that we call amphipathic. When you have something that is part polar and part nonpolar, you're going to have this arrangement where you have the highly ordered waters that are unhappy, they're unstable in a sense because they're so highly ordered, located around the fatty acid tail, the lipid tail of the molecule. If you have a molecule like this, it will want to find other oily surfaces to put together. So if you have 3 of these molecules, their hydrophobic lipophilic tails will associate. They will come together, and they will, in solution, the oily surfaces will join together. And you have all of these waters that are now freed. Well, what if you have, and this is why you will see soap group together and even form bubbles in water. What's interesting about soap, you notice soap bubbles. You notice that soap will join together in water. Soap is an amphiphilic molecule like this fatty acid molecule that we have right here. It's amphipathic. And the soap will actually join together in circles. How can we understand that? If we take this and we extend it, then we can understand why soap molecules form circles. Because if you extend the lipophilic tail, lipophilic tails, yes, if you extend this behavior and you just hide as many tails as you can, you end up with something that will be like a small bubble where all of the hydrophobic lipophilic surfaces are hidden, and all of the head groups, the hydrophilic head groups, are exposed. This is a very stable configuration in water. This is called a micelle. And this is a spherical arrangement where all of the tails of the fatty acid tails of the molecule are hidden. Soap has this arrangement, and that's why it makes spheres or bubbles in water. But the other thing you notice about soap bubbles, it actually will make bubbles with air. And so there are even more structures that can happen. And so one of the things that we observe, circular structures that we observe in life, are vesicles like this. These are actually hollow. They have a thin layer of oily lipid, and inside they have a water solution. This is really interesting because the water solution is kept separate from the main solution by the lipid layer. And this is what we observe under a microscope. These are lipid vesicles. These are made with soap in an orange liquid, and you can see that the soap molecules will form a bubble around the orange liquid. So how do we understand this? Well, this is actually that the soap is forming a circular structure that is not solid in the middle, but it's got a circular structure where it's got more water able to be inside. And that is what a membrane is. A membrane is an arrangement of lipids that has two layers to it. So you have the lipid layer is buried inside two lipophobic hydrophilic layers. The complex structure looks like this. And if you look at this, once you make this sort of spherical structure, the vesicle structure, you can fit anything you want inside, even another vesicle. And so you can have, this is the basis of cells. This is how this stuff works. And this is because water itself is very dynamic. This will result in a lot of other dynamic behavior. So now I'm going to show you a couple more videos that hopefully we can work, you can see how this bubble behavior works. So let me show you this. This is a video on Vimeo where they actually take just, I don't know if you've ever seen the little light sticks. They're a glowing solution, a plastic stick. You break it, you mix the solution inside it, and it starts to glow green or pink. They're very neat, but they don't have much use in Burundi. But they're available if you break them open and put them in water. They will naturally do this. This is basically a simple chemical solution. And I want you to see what it looks like. I'm going to turn down the volume because if I remember correctly, it's pretty loud. But take a look at this. This is just mixing fluorescent molecules, soap molecules, and water together. And a little bit of sodium hydroxide base makes them mix and swirl. You have all this amazing motion. They almost look lifelike. But this is actually not living. This is chemical. This is water pushing these things around, the hydrophobic effect and all those other things. So I'll give the credits for that at beautifulchemistry.net. But that's the idea of what those things, how those things work. I want to show you one more set of videos. The set of videos looks like this. There's basically they show fatty acids, and they show oxygen atoms in these molecules as red, carbon atoms as gray. These are not complex. They're just carbon and oxygen, and of course hydrogen to cap off of all the bonds. But they're basically carbon and oxygen interacting in water. If you have a long tail, you have a fatty acid. If you have two long tails and a head group, you have a phospholipid. The phospholipids will actually gather together to form these hollow vesicles rather than micelles. And the one important thing, I want you to remember this for later. The vesicle has two layers of lipids. You see how it has an outside layer and an inside layer. And inside, in the middle here, you have the ability for another water solution to exist. Water can be inside. The two layers are actually distinct from each other. There is an outer leaflet and the inner leaflet. If you have a vesicle made of phospholipids, it will have this behavior where you have an outer and inner leaflet. If you have a red blood cell made out of phospholipids, you have the same behavior. And so red blood cells also have outer and inner leaflets. So I'd like to show you, that will come into play later, but for right now, just I want you to see how phospholipids naturally associate to form vesicle membrane structures, okay? Here are these two videos, and these again are simulations, but these are simulations of simple molecules that have complex dynamic behavior. And this comes just from having molecules that are amphipathic. They are half hydrophobic, half hydrophilic, and they behave in water as to form compartments. So let me call up those videos for you right now. This is the first one I want to show you. Oh, and let me turn off the volume here because I want to talk to you about it. This is so you can see that this is a vesicle in water. And the important thing to see, it's constantly in motion, right? But the water is not able to cross. You have some motion of the fatty acids, and the fatty acids can move around. You even have some motion from the inner leaflet to the outer leaflet, although in cells that motion is reduced. I want you to watch it again. From the outside, a vesicle looks like this. It's exposing a lot of oxygen atoms that are hydrophilic. But we know that just inside that layer, there is a lipid layer, a lipophilic layer that is made of carbon atoms that are insoluble in water, and the water cannot dissolve and pass through this very easily. So this shows you what a vesicle looks like and how it moves around. I want to show you how vesicles can actually form. If you look at this right here, this shows fatty acid micelles. When fatty acid micelles run into each other, their hydrophilic parts start to want to remain exposed, and their hydrophobic parts want to hide. And that starts to make a long membrane-like structure. So membranes, it's important to see, they are held together by weak, non-covalent forces. They are dynamic, which means they move around a lot, and they also form spherical structures like this. And this happens starting with micelles. This it doesn't take a directed process to do this. This happens naturally in solution. So you can see how those join together and they form. It's actually got its own sort of beauty to it. They eventually form spontaneously a spherical, hollow structure with water in the middle, and they form two compartments. Water, once it forms, there we have it. The water inside cannot cross the barrier very easily. It can only leak out very slowly and if there's a lot of force, because the water and the oil do not dissolve. Okay, I believe that is the videos that I want to show you. Oh yes, one more thing I want to show you. You can actually measure how easily can something cross a lipid barrier. And the answer is, how lipophobic is it, or lipophilic is it? It must dissolve in a nonpolar substance. It must not be able to hydrogen bond. If it can hydrogen bond well, it's going to be happy in water, but unhappy in oil, unhappy in a lipid environment and in the middle of a membrane. So therefore, you can have these different kind of permeability coefficients. What's important for this is just to look at the kind of molecules we're talking about and see how big the permeability coefficient is. And these are also, by the way, these might be slightly different for different types of membranes. Many of these are artificial membranes, which are made like the vesicles that I showed you out of simple lipids rather than the more complex lipids in life. But the general trend is going to be there. Sodium and potassium ions are highly charged. They are not very permeable. They cannot cross membranes. And so that you have very low permeability coefficients. Oxygen has a high permeability coefficient. This is actually good news because it means you don't need an oxygen transporter in a cell. You just let the oxygen diffuse across. And so an oxygen actually has a pretty high permeability coefficient. Carbon dioxide as well, pretty high. Water is actually still pretty high, but this is for an artificial membrane. I believe that cell membranes are less permeable to water than this. The important thing is that charged molecules are very low in permeability. In hydrogen bonding molecules, if you look down here, the more hydrogen bonds you can make, the less permeable you are, even if you're a small molecule that can squeeze through the gaps in a membrane. If you are charged, it is hard for you to pass through the membrane. Steroids are actually pretty lipophilic. So steroids have reasonable permeability coefficients, relatively high, especially considering how big they are. But urea, glycerol, and small molecule drugs are lower. Still not, they still can permeate a little bit. If you have a lipophilic small drug, it has a bit of a chance to permeate the membrane a little bit. It depends on the structure. And the more charges you have on that structure, the less permeable that small molecule will be. This is very important for drugs because drugs have to, when they're moving through the body, they have to pass through membranes, so they have to be lipophilic, but they also have to dissolve in water, so they have to be hydrophilic. How does a drug do that? Well, it can't be all things to all people. It can't always pass through membranes, or it can't always dissolve in water. And I have a story about that with the malaria drugs that we're going to get to. But we're out of time for this right now. I still need to talk about some other things about water before we get to that. And I will end this right here. And then next I will be talking more about what pH is, because pH will determine whether something has charges on it or not, which will determine how permeable it is to a lipid membrane. Okay?